Thank you very much. Thank you, Allison. Thank you all the people that organized the meeting. It's special to be here. 9 years ago, 2013, I gave the keynote lecture at the American Institute for Architecture that had their annual meeting here, as Allison was saying. And what did I say? You said 13. Oh, 2003, right. And the theme of that meeting was called Beyond Intuition, as intuition was a driving force, and the president at the time wanted to make the point that we needed to move forward into more systematic approaches, or make another attempt at adding systematic approaches to the field of architecture. I became interested in this area because 10, 15 years ago, I personally became aware of, in my own laboratory, the plasticity or the structural changes that were inherent in the brain itself, and how experience and environment can change the underlying structure of the brain. And serendipitously, by virtue of some work that I had done, I was contacted by some members of the American Institute for Architecture. In my work, it was in experimental animals, and it showed that the environment had some impact on the brain. And they came to the Institute, called me up and came to the Institute and asked me to come to their hotel room at the Hilton Hotel some 11 years ago, and explain to them how changes in the environment could change the brain, and how they could build things differently so that it could affect the brain. Of course, I had absolutely no idea, but it struck us that we should know. And this is the sort of information that we ought to strive to achieve and to make available to people who are trying to who are designing the spaces that we live in. So that was 10 years ago, and that was the awareness of how brain plasticity was emerging. Since then, we've learned a lot about brain plasticity, but we've also learned that there are other forms of plasticity that occur in the body, and that it's not just that our brains are plastic and changing and responding to the environment, but our whole bodies are, and even our genomes. And I'm going to give you a little series of stories and vignettes about some old and some new evidence for the fact that we are significantly more changeable, variable throughout life than we thought previously. And the basic tenet, of course, is that the brain is the organ that controls behavior. The brain changes as a result of the experiences and the environments in which we live and work. And the designed environments can change our brains and thus our behavior. Do we know enough about this relationship to take advantage of it? All right, the brain is an organ that sits up here, and the model for the brain for many years has been a computer. It processes information. Information comes in, you process the information, it goes out. And of course now there's a greater appreciation for the fact that it's an organ, it's a tissue. It's made up of many different cell types that are constantly in motion, that there's blood flowing through the brain, that cells are changing their shape in response to the environment. But taking into consideration that we're dealing with something like 100 billion neurons, and each neuron then is connected to many other neurons, giving you something like 100 trillion connections, gives life to the idea that the brain is complex. So we're going to break it down a little bit, give terminology. I'm going to break it down further, bring it back out again several times throughout the talk. So a neuron is just one of the cell types, but it's the one I'll focus on. There are as many, if not more, other types of cells in the brain, different types of glial cells that are critical. And here we see one of them, which is called the oligodendrocyte, which acts as an insulator and increases the rate at which information is passed along the axon. So here's the cell body. These are the little antennae, the dendrites that receive information. These little balls there are the points at which the next cell is sending their connection onto this cell. This is the long axon which sends out information and makes connections with thousands of other cells. So the information that's processed and comes into the dendrites is aggregated within the cell body and then communicates that bit of information to thousands of neurons at one time. But neurons are not all the same. This is just a small sampling of the different types of neurons that exist in the brain. Each one of them has a separate function. As you can see, they'll have different size of their processes. They'll have different lengths of their axon and different amounts of insulation around their sheath so that they can communicate information at a different rate. Well, this difference in individual cells is played out in the fact that there are different structures of the brain that serve different functions. So it's not fully distributed throughout the brain, but as in vision, there's certain substructures with pathways that are used for processing information from the eye through the optic tract onto the lateral geniculate through the optic radiations into the visual cortex where the processing occurs, or in-processing occurs. But even in the visual system, that area of the cortex is broken up into smaller subunits, each with specialized units of information, compartmentalizing information, but integrating the information just about vision. That same sort of fidelity exists for the somatosensory system, touching, where in the cortex, along the somatosensory cortex, there's what's called a homunculus, where distributed information is processed about how we interact and how we feel and touch our environment. This is also displayed here in a pseudo diagram of somatosensory cortex, showing the different areas of the cortex that respond selectively to individual touches. We often think of this as a process, a machine. We understand each one of the critical elements of it, but the dynamic part of this comes from the fact that there are what are called critical periods. And critical periods are phases in life in which the organism spans where there's heightened sensitivity from exogenous stimulation that are compulsory for the development of particular skills. A good example is that neurons in the brain which process visual information actually develop after birth. So the way we perceive the world is to a large extent determined by those things that we experience in the early stages of our life visually. Likewise, language. The first few years of life constitute a time period during which language readily has, usually before 5 years of age, has a very important feature. This is exaggerated in the case where certain countries that don't hear, for example, in Japan, R and L before the 18th month of age will never be able to make that distinction. So the brain needs to have these experiences in order for it to be able to respond later. These are called critical periods, and these are the areas of most structural plasticity that occurs in the brain. You can see this as the brain develops in utero, here just from the last 8 months and then right at the time of birth. From that moment of birth on, the brain then expands even further. So it's not just that you're rewiring things, the brain is actually expanding dramatically. From the period of just of birth to these postnatal periods, the expansion of the cortex is really quite impressive, and I must say we don't really understand the nature of that expansion. How much of that is connections? How much of that is increased myelination that continues to occur later in life? The synaptic pruning, and in some cases even birth of new neurons. That's one of the vignettes that I'll tell you a little bit about. One of our heroes in neuroscience is Ramon y Cajal, and he addressed this issue of structural plasticity and how fixed the brain is postnatally, and that once development is ended, the fonts of growth and regeneration of axons dried up, the centers are fixed and mutable. I should say he goes on to say that this is true with regard to the techniques that we currently have, and that as technology develops, we may, in fact, prove me wrong. Well, some over the last 15 or so years, it's become clear that there are areas, few areas, but there are areas of the brain that are do add new neurons even, not just glial cells and connections, but add new neurons over time, and I'll tell you a little bit about that. One of the areas is a brain structure called the hippocampus, which is involved in learning and memory. And one of our speakers today, Larry Squires, is the world's expert on memory, which is probably one of the most important functions for our ability to grasp our environment. So in this area of the hippocampus, which receives information from the sensory and the motor cortex into the hippocampus, where it's aggregated through a defined circuitry, and that information, that olfactory information, the emotional information, visual information, is aggregated into what we think of as a memory, and then that memory then is stored in some distributed way in the cortex for future use. But we believe the formation of that memory of the different sensory modalities and the experiences occurs within the circuit. Well, it's in that circuit, at the first stage of the circuit, where there are new neurons that continue to be born over the lifetime of the organism. It has a really distinct pattern of growth where the cells are initially in a stage of what we call a stem cell or a primitive undifferentiated cell. All of you have these in your brain. And with activity and experience, these cells will begin to grow and send out their processes and begin to make connections. And once they're integrated into the circuit, they can persist for the lifetime of the organism. And as we understand it now, the information that they gather when they're young, just like when you're young, is crucial for what that cell will do later on. So each cell is processing information about its environment. Here's an example of the changes that can occur in synapses. So this is a dendrite, or process with one of the processes that receives sensory information, and these little plugs here, branches, are the points at which information is coming in. This is a young one. This dendrite is only 3 years old, and it's a movie. You can see this is a process that's extending out from that cell. It's a young cell searching, or young spine searching out, trying to find and locate its process. Now fast forward 5 months, and we see what do we see? There you go. Now these spines have now stabilized to a large extent. So this dynamic phase of structural learning. So this is, we talk about learning, we talk about critical periods. They have a physical manifestation that then changes physically over time. So our concepts of plasticity are not just a vague recollection of what happens, but they are reality. We know this process of making new brain cells has a very prescribed sequence of events that occur, and it takes up to 2 months for these cells to fully integrate. And during that 2 months of maturation, just like when you're maturing, there are critical periods for which they require stimulation to achieve full maturity. We know that this type of neurogenesis in the adult brain is something that occurs in all species, from rodents to humans. And more importantly, we know that it's regulated by experience. So environmental changes in the environment, aging, stress, sleep, exercise, even diet affect this particular form of plasticity. I should say I'm focusing a little bit on this adult plasticity because it's such an extraordinary form of it, but it's more of a surrogate for the other kinds of synaptic plasticity that are really going on in the brain in many structures. We know that this structural plasticity in the brain is affected by good things and bad things that can, in other words, you can have a positive impact or a negative impact on this process, and there are consequences in reality. This is a thesis of a student that just graduated yesterday with his PhD, further examining the role of experience dependency in these new neurons. So it's an ongoing process. Here's one experiment that highlights the structural role of the environment in the physical nature of the brain. These are mouse cages where our poor mice are normally housed in this not so enriched environment. If you just put them in an environment, a larger environment like this for 30 days and compare them to an age-matched, sex-matched, genetically identical set of mice in this environment, the number of new neurons that survive by the end of that month is extraordinary. So here's a control. Each one of these black dots corresponds to a newly born cell. This is an animal that had a running experience. They were allowed to run actively in their environment. And here's one that just lived in a separate environment. They had extraordinarily number of new neurons. Normally, the animal has something on the order of 270,000 granule neurons in this particular area of the hippocampus. With just 30 days, they have 317,000, or 15% more neurons in the brain by virtue of this unregulated exposure to the environment. The environment is also, as I mentioned, different types of experiences can have different effects, and even physical movement, as opposed to a more restrained existence, can have dramatic effects. Much to many of ours happiness, this affects not just in young animals and youth, but also in aging. So we know that there's an age-related decline that occurs in many of us, and the question is whether or not changing the environment or changing experience can influence that. One way this is done is to test animals in a memory task where they have to swim around with lots of spatial cues in the environment to find a hidden platform. With some time, they learn this quite well, but older animals often lose this ability to do it, and it's not because they can't see, it's because of changes in their ability to remember what they'd learned in the previous trials. Just having animals, old animals that hadn't had any kind of experience in an enriched environment or physical exercise can increase their capacity to acquire these tasks. This is an example of a study that's been repeated by many investigators throughout the world with a variety of different species. But importantly, not just changes in behavior, but the total number of new neurons in the young animals versus the old can be restored to at least to the level of the young animals and the old animals that had been exposed to these changes in their environment. And it's not just changes in the number of neurons that occur either. The vascularization of the brain changes, and one can see significant increases in the numbers of blood vessels as well as the width of these blood vessels. And it's this change in vascularization that one of my colleagues used as a measure to try to see whether or not there were physical changes that could be detected in the brain of humans with changes in experience. So what Scott Small and his colleagues at Columbia University did was use functional magnetic resonance imaging to measure the changes in particularly the hippocampus after physical movements or physical interactions in the environment. He's not measuring here the birth of new neurons, but using as a surrogate blood flow and changes in blood volume and changes in blood flow in certain regions as a function of this dynamic change in brain function. So first, of course, he took mice and exposed them to changes in the environment, physical exercise, and detected that there was a rather dramatic change just in the structure of the dentate gyrus where neurons were occurring, but only after 4 weeks of exposure, where the other areas showed much less change. So there was a regional effect of physical exercise on one area of the brain. And this was reflected also in the brains of these animals, showing an increase in neurogenesis. And this is correlated with the change in blood glucose. Armed with this relationship between blood glucose and the birth of new neurons, not being able to measure birth of new neurons in humans, he set up a series of experiments with his colleague Richard Sloan to see whether or not physical exercise in humans would have some impact on brain and behavior. So they took a series of couch potatoes and gave them a physical exercise regime over a period of 6 weeks and measured in these patients the brain activity, of blood glucose activity in this hippocampal region during that period of time. And once again, this structure in the dentate gyrus revealed an increase in activity relative to the other structures, and this was correlated with exercise activity. Furthermore, when he tested these patients on a variety of tasks afterwards to determine whether or not this period of exercise had any impact on cognition, he was able to detect a significant increase in performance in at least one learning and memory task that appeared to be dependent on the hippocampus. This is now being expanded quite dramatically in a variety of directions, but a hint at and demonstration how physical movement or interactions and experience can change our brain. I'm going to take you now deeper into the cell, so hold on to your hat or whatever. So we've been talking about neurons and cells at a very global level, but inside, if we look inside the neuron, there's some surprises. The neuron has a nucleus as it's one of its most important organelles. And the nucleus, so cells make up the body, and every cell has a nucleus, and every nucleus has what's called a genome, which is a series of chromosomes that are wrapped tightly together. These chromosomes are made up of what are called genes that generally we thought of them encoding for proteins. This is DNA, and the DNA is a template for making RNA, and the ribonuclear transfer results in small pieces of RNA that act as a template for amino acids. These amino acids then aggregate together to give rise to proteins. In every cell then is making its own proteins that make its cell membrane, that make the skeletal structure that make up of the cell. And this linear doctrine of biology, as was alluded to by John, is the core of life sciences currently, and that is that the DNA is the template for the proteins that are made in the body. And we think of it that way as a linear process, and it's stable. But there were some questions. There's some quandary going on here. Here's the genome, all our chromosomes lined up in their different shapes, their different flavors, and every single cell has a genome. But as I told you earlier, you have lots of different cell types. How is it that one genome, the same genome, makes all the different types of cells that exist in the body? How does that happen? Why don't all of our cells look the same if you have a single genome? Well, it turns out that there is a transcriptional code that regulates this process, and it's through what's called epigenetic modification. There are proteins that bind to the chromosomes, to the genes, that regulate them differentially in every cell. And it is this epigenetic regulation of this code that allows for the differentiation and the patterning that exists. This difference in genomic expression occurs in space. It occurs through time. It can change with time. And the coding and how our genes are regulated can be affected by our experiences, by our challenges, by the events in our environment. So It looks a bit like the details are that mitotically inherited layer of regulation is superimposed on our genome. Very specifically, there are cysteine DNA methylations and histone modifications that mediate or modulate the readout that gives rise to the function of each and every cell of the body. And they're crucial for most of the functions that we have. It's not a linear process. It's a highly regulated process that continues to be regulated over time. So I'm going to tell you a story that relates to this about epigenetics and stress. So researchers have known for years that children whose mothers were chronically stressed during pregnancy by famine, anxiety, death of a relative, or marital discord, for instance, show higher than normal rates of serious psychological and behavioral disorders when they're older. Why? Is this the environment that's changing us in some way? Is this a predisposition genetically? This remains unclear, but recent studies have pointed to a phenomena that I've told you about called epigenetics that may likely give some answers. So epigenetics is a type of gene regulation that can be passed from one cell to its daughter cell. The most common mechanism is through this mechanism I told you about methylation, and the consequence is to inactivate a gene, block a gene's function through methylation. In the case of stress, previous studies have suggested that methylation of a gene, which encodes a gene called glucocorticoid receptor, is very important. Glucocorticoid receptors relay signals from stress hormones to the blood to the cells. In particular, they do so in those regions of the brain that are involved in emotional behavior. So newborns whose mothers suffered from depression while they're pregnant are known to have more highly methylated glucocorticoid receptors. This has implications. So dampened glucocorticoid receptor gene activity has been shown to increase the risk of obesity, depression, and some autoimmune diseases. Why, in light of such negative effects, have humans evolved to be programmed after they're born? Why are we continually programmed by our environment? Part of the answer is that some consequences probably have positive impact. What if our parents' environments change and a mother moves into an environment that's more fearful-inducing experiences? Then giving the offspring this sensitivity to fear may not be such a bad thing. What can be done with such knowledge is unclear. What role can the built environment do to reduce stress in hospitals and homes and hospitals and schools to modify genetics and behavior? Let it be known that is happening, whether or not we are doing it actively or not. We are changing our genomes, not just our brain. So let's dig deeper. You're not asking for this, but I'm giving it to you. I've told you about epigenetics. Now, what about genetics? I mean, the genome must be stable, right? That can't be changing. But it turns out now we know that the genome is not stable. The DNA itself is not stable. Parts of the DNA move around. Parts of your DNA move around in your cells. These are called mobile elements. They were first discovered by Barbara McClintock in the 40s, met with significant amount of resistance, but eventually in '83 she was given the Nobel Prize for this discovery. It was discovered in maize, in corn, but it underlies the differences in phenotypes that exist between different corns. And in her discovery, she found that pieces of DNA make enzymes that clip themselves out and move themselves to other parts of the genome. Well, for many years, we knew about this, and we sort of resolved to think about this as some sort of junk, some residual. It was called junk DNA, or the selfish gene. Richard Dawkins wrote books about how this was not, couldn't be important. This is just something residual left over. But then as the genome was sequenced and we began to understand something about the genome, we had to have a second look at this. 2%. This is this is the total amount of DNA bases in the genome. 2% of it makes protein. Remember the central dogma? DNA makes RNA, makes amino acids, makes proteins. That's 2%. That's what the genome does. This is all the rest of the genome. 50% of your genome, 50% of every cell in your body is made up of sequences that represent mobile elements, either historic or currently active. The ones that we've been interested, these are all parts of this archaic family that's been with us over time. And more extraordinarily, they have a very specific pathway at which they function, where one particular type of transposon is called a retrotransposon because it goes through an RNA intermediate. Instead of just cutting itself out here and knocking itself to another location, it makes an RNA intermediate and then produces proteins itself, which can nick the DNA and insert it randomly into the genome. We and others now have shown that this occurs in the brain during neurogenesis, during development and during other parts of even in adult neurogenesis, where neurons can be marked for those cells that have had a genetic insertion, a novel genetic insertion, which appears to be, as much as we can tell, random. Now, evolutionarily, you'd think that this should be going away, right? Well, this is the amount of mobile elements that exist in species over time. We're gaining. Our genomes are increasing in the amount of mobile elements that exist in the brain. And this is supposed to tie things up, if you're following any of this a little bit, this is supposed to tie it up a little bit. It turns out that methylation, which is one of these forms of epigenetics, is a strong regulator of line activity, of mobile element activity. It suppresses. Remember, epigenetics and methylation is a suppressor, like in the glucocorticoid receptor. Well, methylation, it binds in this area here and blocks this line element from being active. Now, MECP2 is epigenetic mechanism, and its function is to suppress genes, and it suppresses genes in very effective ways and highly modulated. What's the impact of not being able to suppress these genes? What if you couldn't? What if you were misregulated in your ability to regulate suppressed genes? What would happen? What would happen with lines? Well, it turns out that there's a disease called Rett syndrome, which is a form of autism, which has a deletion in MECP2. They don't have MECP2. They cannot suppress the genes that MECP2 normally methylates. And as you know, this autism is a childhood developmental disorder, large spectrum of disorders. This is a unique one because we know the gene that's involved, but the cells are small, the dendrites are small, the connections between the cells are small. We're beginning to learn about them, about this disease, but it's still, quite frankly, a serious mystery. We explored whether or not this lack of MECP2 in the brains, in the entire body of this organism, had any impact on the mobile elements in the brains of patients with Rett's. And the way we did this was to take skin fibroblasts, this is a whole weird technology, grow the cells in a dish, transfect very specific genes in your own fibroblasts, and we can convert your cells into embryonic stem cells. Those embryonic stem cells then can be induced to become neurons, which are replicates of the brain cells in your brain. And this is a technology called induced pluripotency, which was discovered by Shinya Yamanaka about 5 years ago. It's a burgeoning technology now where we can take cells from anybody in the audience, grow your cells, revert them back into an embryonic stem cell and make them into neurons and measure your neurons in a dish. Or your heart cells, your liver cells. We had this opportunity, working through the Rett Society, to get cells from patients with Rett syndrome to see whether or not when neurons are being formed, is there any change in the mobility of these line activity in these subjects. So we took the cells, reprogrammed them into embryonic stem cells, and turned them into neurons through a process that's now becoming more standard. And to our surprise, using this as a reporter construct, we saw nearly doubling of the amount of new insertions in the brain cells that were derived from the patients with Rett syndrome relative to the non-affected patients. In another set of experiments, we took autopsy tissue directly from the brains of the Rett kids and analyzed, using DNA sequencing, the amount of line activity on a per cell basis, and found that in fact they also showed an increase in the amount of DNA in the brain relative to the controls of these subjects. So we are beginning to think that under normal circumstances we have these cells that are really under tight control, silenced by these epigenetic phenomena, that during normal neural development there's a release of some diversity generator, some movement of these cells that randomly insert themselves in the genome. And this can even occur in adults, where we run the animals in a running wheel, increase neurogenesis, and there's an increased amount of line activity that occurs even in the adult. So we as individuals have multiple stages during our developmental pattern where we are differing from that which originally occurred through the merger of the sperm and egg. So even at this early age of the zygote, there's motility, transient motility in the genome, certainly at the blastocyst stage, during neurogenesis in embryo, and during neurogenesis in the adult. So we believe that this individuality that is being generated by these mobile elements is leading to a broader diversity in the environment, broader diversity in behavior. Our proposition is that you are unique. Not only are you produced by your genes and your environment, but you're also a product of chance. So built on top of all these areas of plasticity, we are in fact individualized, and how we act in the environment will be in some sense determined a bit by ourselves as well. So we're not all the same. And even when you're designing the environments, when you're building our buildings, we need to take into consideration the fact that there's severe individual differences. So genes provide a general blueprint for the organism, and the environmental stimuli influence many characteristics of the organism. Environmental stimuli act through regulating genes to influence organisms and its behavior. This last vignette came to me, I wanted to share with you. I had a student that came and applied for a postdoctoral fellow at my lab, and I asked her if I could show some of her slides, because they struck me as remarkable, and it's changed how I sleep at night. So this is light patterns. This is work by Tracy Bedrosian at Ohio State. And this is the middle of the night, showing how much light exists throughout the Earth. You can see that some places are really quite dark. And that there's growing appreciation that circadian rhythms or sleep cycles are having an impact on mood and brain plasticity and immunology for sure. So cycling rhythms are a 24-hour oscillation, affected by sleep and arousal, reaction time, body temperature are all regulated through this cycle that occurs through a 24-hour period. So you have light is the input. It modifies the oscillator, and an oscillator affects our behavior and our structure of our brain. It's all mediated through your eyes. So the light comes in through the eyes. There's a very selective set of cells called the melanopsin cells in there that are separate from the other retinal ganglion cells that are functioning through the optic chiasm to regulate these dramatic patterns of brain function. And there are specific wavelengths, and the blue light, for example, is much more effective in regulating our circadian rhythms than these red sequences, which are much less, and something actually for you to consider. All of this is centrally located right here. If you damage this structure, you lose your ability to cycle. And on top of all that, a lot now is known about the proteins that are involved in cycling. So there really is a clock internally within these cells. So the proposition is that circadian disruption alters mood, and that mood can also disrupt these circadian rhythms, and that mood and circadian rhythms are certainly affected. In modern civilizations, disruption is pervasive and understated. 99% of the population exposed to light pollution. So 24 hours a day we get all sorts of light exposed. Here are the changes that occur in light just from the 1850s up to 1997, and this is the projected midnight night cycle happening. So here's this last bit of relatively speaking, shift work has the consequences, and there's an interesting series of studies on the Amish who maintain no electricity and have very, very remarkably low rates of depression. So many investigators now are beginning to look at this role of light and circadian rhythms on our behavior through these brain structure activity. And just one example with this wonderful little creature, the Syrian hamster, which has a very defined cycle, very much like humans. In this one set of experiments, they kept the animal in dark for 18 hours, in light for 16 hours, and in complete darkness, 0 lux for 8 hours. And the experimental group had just 5 lux. So what's a lux? So a lux would be 1 lux would be like full moon at the tropical latitudes, and we're talking about 5 lux. So this is, a low light family room would be 50 lux, so a tenth of that. And what she showed, there are a variety of studies on this now showing changes in inflammation, changes in brain structure, changes in behavior in less than a month of this low light exposure during sleep periods. And it's manifest once again to our very happy spine. So here's apical and basilar spines of animals that are exposed for just 1 month to this low dim light. So count the black dots. This is explained, so this is how clear all of this is. It means we have lots more to discover. And I'm restating this a bit, but I'm also saying a sharing of knowledge between neuroscience and architecture may help us design buildings and spaces that are more optimal for tasks and performance in those spaces. And you saw yesterday what I think is probably the first real effort to bring together the evidence-based approaches to understanding the interface between neuroscience and architecture. And that's this remarkable group of folks that have gathered together here in San Diego to look at using Cal-IT. It's a collaboration led by some of our key players, as you can see. And this is some of the work done at Caltech. Here are some slides that Eduardo gave me of the happy campers that are experiencing these virtual environments with their headgear, so you can monitor the brain activity while they're wandering through prescribed environments, measuring all the different things that are giving us the beginnings of how changes in the environment, change in our function can affect our behavior and emotion. I'll leave you with this picture of the Salk and this picture of Louis Kahn and the Salk, who argued a lot about the building. I can talk about the stories, and many of you know about how they wanted to have, Louis Kahn wanted to have 3 buildings instead of 2 buildings, and Salk didn't want it. But why is it that the Salk is so effective, if it is? I think it's a great place. I think it's a beautiful place, but it's also a place of great productivity for some scientists. And what is it? Is it because it's such a beautiful place? Is it a great setting? Well, one of the things that's most remarkable as a person who actually works in this building is that these concealed trusses that exist within a building, here's 1 floor in the Salk, allows for unobstructed lab spaces. So in every space where we have labs, if you take all the benches out, this is where it is. There aren't any walls. There's no hard walls in there. So we can redesign our buildings And I've been here many years, and almost every year I'm putting a new wall up here or a new place there. And is that maybe the reason why this is effective? It's not because of its beauty, because the design was done in such a dynamic and plastic way that allowed us to get through here. Well, I don't know the answer to that, but those are the kinds of questions hopefully we're going to be addressing and have been addressing in our society. So here are my collaborators, scientists and architects, and we are going to find the merger here at the beginning. Thanks for your attention.
Thank you, Rusty. For those of you, are you willing to entertain some questions?
Sure. If you're willing to ask.
For those of you who may be feeling that you'd like to watch this presentation again, just to have it sink in, and maybe again and again and again, we are recording everything at the conference. Eventually we will be posting the proceedings in some manner. I can't tell you at this point exactly how, but previously we've posted our lectures on our website. So we have some runners with mics, and if you have a question, please put your hand up and the runner with the mic will come to you. And this advice goes out to all the presenters too. Because we're recording, please make sure you speak directly into the microphones. So, any questions?
That'll intimidate them. Yeah. Here we go. Mike. Michael with me.
So the tough question is, do you think architects really need to know about epigenetics? It's clear that understanding brain plasticity in terms of, let's say, the hippocampus can change with certain experiences, we can do certain things to optimize those changes in terms of the needs of the architect or the needs of the inhabitant. That's clear. How do you see the pathway from all these wonderful details of genetics you've shared with us up to the level that the architect needs to know about?
I think that's a good question. I think I would refer back to John's discussions of consilience on E.O. Wilson's book, which is one of my favorites. And I think that first of all, we don't know. I don't know what the difference is. But you wouldn't want to hide information. You wouldn't want to not present information that were available at all levels. You don't want to dumb it down so it has some, and I don't mean that in any derogatory way, you don't want to just present some aspects of it that you're comfortable with. You want to present the stuff that you're a little uncomfortable with and let people think about it. Let people bring to bear what it might bring, the impact that it might have on your decision making. And it won't matter for everybody, but it may be a few people that find that information somehow translates for them better what it is that they mean by the structural interface between the two. And more is better as far as I'm concerned. And as sort of the scientific provocateur of this group, it's part of my challenge to bring the knowledge to you, where the cutting edge is on it, and use the knowledge as you can. This is only today, and just this information is less than 10 years old, the new information. And 10 years from now, this will be old information, and there'll be a whole nother layer of information that's much more complicated. So we need to get to know this knowledge. We need to be aware of it. We need to integrate it into who we are and how we think about things. We can't be stuck thinking about things as they were. That said, everybody has got to find the comfort level zone and stretch as far as they can for what they can get at that moment. But I think we need to keep plowing through it. This is called hand waving, but it's okay. He knew that was coming. Yes.
Two maybe separate issues. One is that on the work on epigenetics and transformation, there's a whole, I don't know what to call it, social movement, broadly called somatics, where you think about the body in connection to the mind and in connection to culture simultaneously. And it's pretty experiential, and you learn how to go in and become a dendrite or become the nucleus of a cell. and perhaps even interview it and blah, blah, blah. There are connections with much older meditation traditions. And I'm thinking that that's an area that yet needs to be explored and integrated into our thinking about neuroscience and environment. Maybe related, my second point, is that some of the things you've described, like the advantage of sleeping in the dark, probably come from Vastu or Feng Shui or any other traditional norms for building. And I think that's another domain that needs to be explored and integrated with our contemporary thinking.
Great. I'll respond to that by saying I agree that there are many disciplines that we need to bring into it. One of the things I think that is paramount to our merger of neuroscience and architecture, and the part that I found one of the parts I found most interesting is in the cultural differences that neuroscientists are scientists, so we're empiricists. And there's a real pathway that we have to go through before we can add information into our deck. And it's the experimental process. So we need to gain the information in the environment, we need to generate a hypothesis, we need to get an experimental group and a control group, and we have to do an experiment. We have to test the experiment, and we have to see what the outcome is, and check the outcome based on our original hypothesis to determine whether or not to reject the hypothesis or accept the hypothesis. And from that point on, you can take that kernel of knowledge, build it out to a little bit of generalization. How generalized is that? So these are the methods that we use to try to formulate our neuroscience society. And I think when we incorporate other disciplines into it, we want to broadly accept them, but put them to the same kinds of tests to see whether or not they fulfill the obligates of the scientific methodology. And I think that's been a fun discussion that we've had over the last 10 years, is how do we do that? How do we do experiments at the interface so we can put the knowledge in there as a core knowledge?
Relative to that, it just occurred to me that it might be good or useful to apply the neurogenesis kinds of research that you're doing with mice to wayfinding, which could be measurable and has a direct overlap between architecture and what you do.
Yeah, no, what we'd really like to have is better imaging tools, and that's why I'm so heartened by what Eduardo and his team are doing at the Cal IT2. Is being able to, what you'd like to do this is in humans, and you'd like to be able to monitor over time the chronic changes that occur in the brain. I think we're getting there. We're not there certainly yet, and this merger of the things that we can do experimentally, where you can doing invasive kinds of work in experimental animals versus what we can do in humans, we're still at that merger point, but there's 100 experiments that I think many of us would really like to do if we had the tools. So a lot of it's going to be tool building, and I think we need to we should have more imaging people here at this meeting so that they can understand how important it is for us to do it. I agree with you entirely. Wayfinding is one of those pieces that puts it together.
There's a theory that maybe our brains aren't capable of doing everything we'd like to do. I have a dog who's, I think, very bright, but I can't teach that dog physics because that dog doesn't have a brain that's capable of understanding physics. Do you think it's possible, unfortunately, that our brains are not going to be able to handle the consiliency of architecture and neuroscience?
Yeah. Well, there's another way of looking at this, and maybe that consiliency is not something that everyone is going to have as an epiphany at one time. But what may happen is that there are unusual individuals that can understand elements of the consiliency as an epiphany, and they have a unique ability to communicate what that looks like, what it feels like. And those people can be the pathfinders that then communicate that information to others. Now, we think about this in terms of, the analogy for me is when I think about these unique events that occur in evolutionary history and in cultural history as well. One example would be in art, where Picasso and Braque somehow came to Cubism as a form of art. That, you wonder how that came about, and there probably is lots of literature written about it. But after it happened, and people saw it, and they felt the impact of it, it was easily translatable into other people that modified it into a whole other things. But it took somebody to make that event, to have that happen, and then it can spread. So maybe what we're going to search for is not necessarily everyone getting it simultaneously, but a few of the key people in the audience and beyond understanding enough of the key principles to communicate those so we can incorporate those and move on from there. So your dog may know enough physics. You can teach him enough physics so he can learn certain things, but maybe not understand all the fundamentals of the math of physics, but he can use the principles. So we can be the dogs of the consilience learners.
Dr. Gage?
Yeah.
Regarding enriched, in the back, hi. Regarding enriched environments, as you know and you've discussed, sometimes enriched really kind of just means bringing things back to normal.
Yeah.
And I wonder if there's a possibility that people might misinterpret your work and overly complicate environments in the hopes of, quote unquote, enriching them, not realizing that really in some cases the control animals are kind of living in what we would consider a deprived environment.
Yeah. So the other key to, that I can go into, even in the big environment, the bigger environment, within a few days, if you keep it the same, the animals actually stabilize to it, they habituate to it. So part of the key to the enriched environment is changing the environment on a regular basis. That's a real feature. So didn't get a chance to elaborate on it, but you're very right. And so it isn't just complexity. It's really variance. It's change that in those sets of experiments that are more important. You can even take a smaller cage and move things around, and that has quite a bit. But the other part, of course, is one of my students is now leaving sort of the experimental animal phase of things and wants to try to translate this into humans. So what is an enriched? How is he going to enrich somebody and then do imaging to see what the impact of an enriched environment is? And he's actually coming to the idea that a simpler strategy may be to have it would have to be some unique event that would occur, a trip to a very extraordinary location, a travel trip to someplace they'd never been to before, and or learning an instrument or something rather striking. So you're absolutely right. When we think of getting out of our experimental setting and moving into the real world, like we're trying to get the architects into a smaller world so we can look at the details, but we still have to get back out into the real world to see how that happens. And this is where the dialogue, I think, is most important for all of us.
Is the mic on? No. It is? Can you hear me?
I can, but I can't see you. Okay.
You may have just answered this question, but I'll ask it anyway. Pushing this question of consilience and what the relationship and interaction is between neuroscience and architecture, I come to this discussion from the direction of architecture and environment behavior research, which is clearly being enriched by what I'm learning about neuroscience. And I'm wondering what the neuroscientists are learning from the architects, if you could speak to that.
Yeah. Well, I think you during your couple days here, you will I hope you'll take advantage of that and meet up with neuroscientists and ask them that very question, because each person, I assume, will have a very different response to it. Personally, I would say that the awareness, my awareness that the built environment, every built environment that I find myself in has an impact on me as an individual. So I am being changed by the environments that I live in, my home, the hospitals, amphitheaters, the structural, the physical structure. I know that this is changing or participating in the change of my behavior. And I don't see it so much that I'm learning. I want to participate. I want to have some role in helping to enrich the general field so that we can use principles about neuroscience. So you can use principles about neurosciences, but we can help in figuring out how we can take advantage so that we can build an environment that optimizes the performance of individuals in that environment. So not generally, but for that environment. What do we think that this environment is supposed to achieve? And I think we have to consider who the people are that are going to use it. We have to consider all these variables and what the impact that will have on their structure.
So is what you're learning about architecture changing your neuroscience? That's what I want to know.
Yeah, I would say that I think about it more in terms of how you could ask questions. And also I think, what is the right question? For scientists, we sort of, hopefully before we start doing experiments, we think about trying to come up with the right question and then frame the experimental design around that, that question that has an answer, that's solvable. And I would say that's what we're struggling with now. That's the interface between neuroscience. The folks that are trying to work at this interface are working together to figure out what is the information that we do know and what are the most important questions. And importantly, what are the questions that we actually could get information about? We have lots of questions that we might want to know, but they're not knowable with the current technology. So what can we know? And let's design some experiments and try to build a base of knowledge, and from that base we can expand it from there. It is definitely a teamwork. There's no question about it, that I don't think that the neuroscientist is going to be able to do this. And I believe that the architect is going to need this knowledge to expand. So we're playing a role as teachers or helpers or guides for this new body of knowledge that might be useful in design. Yeah.
This would be the final question.
Appropriate.
Just to respond to that, I think that what we need to do is to find common points, and wayfinding has been brought up as something that is innately interesting to both the architect and the neuroscientists. Neuroscientists are interested in how we use information in order to create maps, in order to be able to see ourselves within our context and to create pathways. And we try to understand how the brain functions in order to optimize that process. And of course, the architect is interested in the external manifestation of that process in terms of the design of a facility like a hospital where people will not get lost and contaminate everybody and so on. So that's why I think we focus a lot on wayfinding, because it's a common point where we intersect. And there may be many others. And so that's, I think that's where we start going towards each other and then out to our own practices.
Thank you, Eduardo.
If someone had handed me the microphone, could I get one more question in?
Sure.
Please make it brief.
Yes, I'll make it brief. I'm trying to understand, not being a neuroscientist, the scope of neuroscience. For example, I've read about some of the research, particularly done by the Heart Math group, on the ability of the heart to essentially have some of the dimensions or the behaviors of a mind. We're now learning about the tremendous role of bacteria, that there are 10 times as many bacterial cells as there are other cells. So before I start to link neuroscience and architecture, is neuroscience related just to the brain, or does it encompass other organs of the body and the relationship thereof, et cetera? So is it useful and helpful to look at those aspects as well in terms of its relationship to behavior and architecture?
Absolutely, absolutely. The brain is a regulator of almost all the structures, and all these structures, like the biome that you're talking about in the gut, processes the food that gets into our brain, that nourishes our brain to make it function. So any representation that I had that it's a single structure independent of the body was mistaken. So yeah, good point to be raised. The body is an organ in and of itself. It's an integrated organ with lots of functions. The brain, however, is the processor which regulates behavior. The behavioral output is regulated by the brain. And so that's why we sort of focus on that. And the surprising aspect of it is the brain is plastic. So it's changing by virtue of lots of things, including the structural and built environment. And that's why the emphasis has been there. But you bring up a really important point, that the brain is part of a whole organism, whole body, and there are lots of changes that are affecting the brain, and the brain affects them.